The overall goal of the DOT assay is to assess the migratory phenotype of attached, cohesive cell sheets in response to microenvironmental cues.
This method can help answer key questions in the cancer field, such as how cytokines or growth factors alter tumor cell motility. The main advantage of the DOT assay is that it is easily performed and yields reproducible results. Though this method can provide insight into cell motility, it also lends itself to a range of subsequent assays, such as staining of cells to assess cross-colony morphology and cell scattering, immunostaining, protein and DNA or RNA analysis, or injection of cells into animals.
To begin, thaw mouse collagen 4 on ice and dilute it with 50 micromolars HCl to prepare 3 milliliters of a 10 microgram per milliliter collagen 4 solution. Add 250 microliters of collagen 4 solution to the wells of 12-well glass bottom plates and place them into an appropriately sized, tight-closing plastic box. Place wet paper towel over and around the plates to manufacture a humid chamber and close the box. Then, incubate the plates overnight at room temperature. The next morning, to remove non-absorbed collagen and buffer, use deionized water to rinse the plates twice, directing the water to the edge of the plate well rather than the edge formed by the plate bottom and the cover slip. Air dry the plates in the laminar flow hood and use them immediately or store them at 4 degrees Celsius for up to 5 days. To prepare cell suspension,